Okay, I think we'll make a start. Hi everybody, for those that don't know you, my name's Ben Markman. I'm a medical oncologist from Alfred Hospital and from Cabrini, and I'll be hosting today's webinar. I'd firstly like to acknowledge the traditional custodians of the land on which today's presenters are located. I'd also like to pay respect to the elders, both past and present, of the Kulin Nation, and extend the respect to other Indigenous Australians present online. So thanks everyone for taking the time out of your busy schedules to join us for today's symposium, which is titled Clinical Application of Genomics, a Practical Guide for Oncologists. This event is the culmination of a collaboration between the Monash Partners Comprehensive Cancer Consortium, MPCCC, and the Victorian Comprehensive Cancer Centre, VCCC. And we've got a fantastic program today that's going to be delivered by members representing these two major cancer networks. We are all aware that genomic-based assays are increasingly being used to guide clinical decision-making to select the most appropriate therapy for any given patient. And our program is going to walk through various elements of that, from the basic science and genetics to a rundown of assays utilised, oncogenic drivers, and some practical elements like patient consent and practical examples of the types of reports that come across our desk that we have to interpret. This will be followed by a panel discussion and a Q&A session. Just from a housekeeping perspective, if you have any questions for today's presenters, you can do this via the Zoom Q&A function or chat, although it's probably easier if you keep it to the Q&A function. You can also raise your virtual hand if you'd like to speak to the panel during the Q&A session, in which case, if we have time, we'll unmute you and you can ask your question. We just ask that you mute yourself again after speaking. Even though we're going to hold off questions really to the end of the Q&A session, because we've got quite a tight schedule, you are free to submit your questions at any stage along the way. So really, without further ado, I'm going to introduce our first speaker, and it's a great pleasure to introduce Professor Melissa Southey, who is a molecular geneticist. She is Chair of Precision Medicine at Monash Health and Director of Monash University's new Biobank Victoria initiative, and Research Director of MPCCC, who's known for her work with multiple case cancer families and heritable risk factors. So Melissa is going to talk to us about the basics of genetics. So thanks very much, Melissa. Please take it away.
Thanks, Ben. Do my slides look okay from your perspective?
Yes, absolutely. Thanks.
Terrific. Well, I think I've got the task of really introducing my colleagues who are going to take on the heavy-weighted stuff in the later part of this session. So I thought I would start with a little bit of history as well as some basic genetics. And this, of course, is Gregor Mendel, who's really regarded as the founder of genetics. He was an Austrian monk who had particular attention to aspects of his garden, including his peas, which instigated a lot of work which he recorded around Mendelian inheritance. A lot of his work was actually forgotten until about 1900 when it was reinvigorated, but he's really regarded by almost everyone as the father of genetics. So if I just move my slides properly. Are we on to the second slide, guys? Having trouble seeing.
Yes.
Excellent. Okay, so some really basic principles, I suppose, and at least until recently, so I guess I want to say that caveat, at least for probably the last 30 years, there's really been two silos of discussion around genetics, and one has been under this umbrella of germline aspects, and the other has been under aspects around somatic, so acquired during life, genetic alterations. And really there's been a big divide between these two aspects. The germline aspects have been around, I guess, what Mendel founded around heritable factors that predisposed one to a phenotype. And I guess tonight we're talking about predisposition to a disease, and particularly cancer. And this has really blossomed as a discipline in itself, and has in the last 2 decades made amazing strides around identifying genetic factors that are heritable and associated with increased risk of disease. And on the other side, similar great strides have been made around understanding the genetic sequence of events that drive a normal tissue to a neoplastic tissue and then into a metastatic, a cell with metastatic potential. But I think some of the aspects that will come through in the talks to come is that this line is gradually being eaten away because the two disciplines are really coming together. There's been a lot of cross-pollination of ideas, both at the basic science level and also in clinical practice, how these two aspects of genetics can inform each other and improve the identification of important genetic information both in tumour tissue and in our germline, and also how it can help both manage risk of genetic disease and also inform clinical management of people at risk of cancer and also who've been diagnosed with cancer. So just to go into history just a little bit, to put you into a little bit of context of how quickly this field has really evolved. This is Edwin Southey, and this has moved to about 1970, where there were really crude methods possible for extracting DNA from tissues, including blood, which were increasingly being used to examine this idea of DNA risk factors, or at least identifiable parts of the chromosome being associated with heritable disease. And Edwin was the first really to describe this process of extracting the DNA, digesting the DNA quite precisely using restriction enzyme digestion, which is probably something an undergraduate wouldn't really hear about these days, but certainly it was the key feature of all genetic analyses for about 20 years, being able to digest DNA very specifically around very specific DNA sequences, which enabled the iconic southern blot to be produced on a nylon membrane, and then for very large fragments of DNA, usually using radioisotope to identify fragments within complete nucleus worth of DNA to isolate and identify aspects of DNA of interest. So this was used extensively across all sorts of medical research contexts. And just to give you, sorry, I'm just having a little bit of trouble changing my slides. Here we go. One context that was used at the Children's Hospital during the 1980s and 90s was, it was really provided a tool to start identifying individuals predisposed to disease, but in the context of the example here, retinoblastoma, the explanation for the cancer arising. So here on the right is a southern blot. The red marks, like the previous slide, indicate areas that have been identified by the probe. You can see in the family tree here that we have a father, mother and 2 offspring, where the father and one of the offspring has a very distinctive pattern on this western blot, which is indicative of the portion of the DNA that carries the retinoblastoma gene and probably carries something like a mutation in it to be linked to the disease occurrence in the family. So it was fairly rudimentary in today's eyes, but at that time it was really, really, really helpful to be able to identify within families who are carriers of the likely allele that was responsible for the disease predisposition, and was really the beginning of managing these families in all sorts of contexts, not just retinoblastoma as far as their genetic risk. But fairly crude, and all sorts of caveats around these sorts of information, but nonetheless it was sort of the pioneering work that enabled the field to grow. This is another, and again it's a 1990, this is Fred Lee and Joe Fraumini, who described the Lee-Fraumini syndrome. This is the description of families who carry germline pathogenic variants in P53 and were most identifiable via pedigrees like this. pedigrees where sarcomas were common in young people, where breast cancer was common at early age at diagnosis in female members of the family, and often there were brain cancers and other very distinctive features of family that were helpful to identifying P53 germline mutations as being responsible for these sorts of family pedigrees. So again, a very large landmark piece of work that helped extend the field and push the field forward. But also in parallel to that, of course, was the improving genetic technology. So instead of having very large cosmid preps with very large fragments of DNA in them, became possible in the early 1980s, I suppose, mostly due to the onset of the PCR reaction. We became much better at being able to sequence very specific fragments of DNA. This is actually a standing agarose gel with, again, 32P labelled nucleotides in it for autoradiography. You can probably imagine this was fairly laborious to read, but this was state of the art in the 1990s when, for instance, this is the BRCA1 gene when we started to sequence BRCA1 in families looking for genetic variants, which you can see is identified here in one of the individuals part of the analysis. So again, not exactly what we'd expect today, but was part of the evolution of the technology. This is actually the Diagnostic Molecular Pathology Lab at Peter Mac, 1980, 1998. You can see the electrophoresis standing up there. This was state of the art, amazingly laborious, amazingly expensive, but did provide some answers and really did provide some momentum to what we know today as genetic analysis. So this did improve, of course. Some of our machinery improved to take us away from radioisotope. We used fluorescent tags. We got chromatograms rather than having to do radiography. And of course, today we actually have next generation sequencing, which is no longer really the next generation, but massively parallel sequencing in a broad sense has provided amazing opportunities for low cost, high throughput, large number of analyses, data beyond what a basic biologist would have imagined 30 years ago. And I'm not going to say very much about the data that comes off this machine. I mean, it does come off electronically. There is an amazing amount of work gone on in the context of managing this data. It's a little different when you're considering germline versus the somatic space. Highly sophisticated, incredibly rapidly developing, will develop for a lot longer and provides the basis of most of, I imagine you will hear in the talks to come. So one of the key attributes of advanced technology is that we can look at the whole genome if we want to. We can sequence just the exome if we want to, and we can also do targeted panels. So just look at some genes in some disease contexts where there's information to think that there are some genes worthy of looking at more than others. So the expense is proportional. In general sense, the less of the genome we look at, the less expensive it is. And this, in the last 10 years, has really created an industry in gene panel testing. And this is just a screenshot from one of the commercial providers in the US on two of their gene panels that they provide for service. And this really has expanded what we understand to be a cancer susceptibility gene, and it really has created data beyond really what we can interpret, which has become a problem. So everyone's been very enthusiastic and added a lot of genes to their panels, even when there's not a lot of evidence to suggest that those genes are associated with disease, and specifically for the disease perhaps being part of the indication. So one of the real current challenges in genetics is how do we classify the variation that's identified using gene panel testing? And there has been a lot of international work addressing this. There's been an amazing effort by the National Cancer Institute in the United States, who have funded a infrastructure, I suppose, referred to as ClinGen. ClinGen has a very large number of gene-specific working groups So for instance, there's a working group for BRCA1 and BRCA2 that solely look at genetic variation in BRCA1 and BRCA2 and work to robustly classify variants identified in those genes. In some instances, the working groups are looking at a number of genes, but in many instances, it's just a single gene because of the expertise required on a gene per basis to classify the genetic variation identified. So that's a very large and ongoing piece of work, which I think will be ongoing for some time, particularly as we work beyond our understanding of European genetics into other populations that are important for us in all sorts of contexts, including providing adequate clinical interpretation in the Australian context. So we have a couple of other activities in place to try and standardise clinical delivery of this information, and certainly in the cancer genetics space, ABQ has been used very extensively. This is just a screenshot from last month. This is a site that's used as a reference point for best practice guidelines, and you can see just by this screenshot that it's being used to introduce all sorts of new information about risk management related to specific genes, and also to update prior information. I mean, in this context, there was a very large publication about PALB2, the guidelines were updated and then released again on this site. So it does provide some consolidation of information and hopefully some harmonising of interpretation of the information around clinics, at least in this country. But of course, as I said, this divide really is beginning to become much less clear between somatic and germline. So just to use the example of this woman here, in a BRCA1 pathogenic variant carrying family, this woman is obviously at increased risk of developing breast cancer, and she can be afforded risk management strategies via clinical genetics. But if she were to develop breast cancer, we do know that this breast cancer is highly likely to be of a specific site. This is likely to be a very high grade breast cancer with pushing margins, often of a medullary type with a really high mitotic index. So the information about the germline actually helps inform how the treatment might be chosen and the ongoing management of this woman and also her family. So there is much more interplay in that direction with genetic information and in movements to mainstream activities in the future, which I think will probably be part of the conversation at the end of the session. So just before I leave germline aspects, I just really can't talk about germline, at least in complex diseases, without mentioning apologenic risk. So just to use the BRCA1 example, here's the BRCA1 curve. The average woman with a BRCA1 pathogenic mutation has a life, at least till 80 years, a risk of developing cancer of just below 80%. But it all has to be taken in the context of the underlying polygenic risk, which does modify the individual's risk and is becoming an important part of genetics in oncology. We aren't just looking at single gene, single pathogenic variants, single disease susceptibility. We're talking about very rare variants on top of an individual's a whole genome sequence, essentially, that all interplay as far as risk of disease is concerned. So a very complex area. A lot more work to do, to understand how much of this actually is applicable to in the clinical context, but quite an exciting area and one that offers much more precision around delivering information about germline genetics. So then just to switch to the other side of the divide for just a moment and looking at somatic mutations, I think many of us, just to come back to my title in Genetics 101, mostly when we talk to young students about this, particularly in pathology streams, we relate this back to the histopathology. So it's been clear, and then they set some fantastic examples to show students how the underlying genetics can really come through in histopathology. So this is follicular lymphoma, a fairly classic example with a translocation between the immunoglobulin heavy chain and BCL2 that drives constitutive expression of BCL2 and makes these amazing germinal centres that are very characteristic of follicular lymphoma. But that's just perhaps a very clean example and very high level comment about follicular lymphoma. And there has been a lot of great work over many decades put into understanding the tumorigenic pathway. And this literature begins back in the 1950s, really before there was any molecular methodology that could really address this issue very specifically. And one of the landmark papers in 1990 was Bert Vogelstein and Eric Fearson's work around looking at colorectal cancer tumorigenesis, which remains a fantastic and fairly robust model today to talk to students about tumorigenic pathways. So he, with hardly any molecular technology capacities at all, was able to string together what he could see under the microscope as far as histopathology was concerned, with some really fundamental landmark molecular changes and genetic changes in the cells. So he, in his paper, walked through the hyperproliferative epithelium and then talked about acquiring a p53 mutation in an intermediate adenoma through to acquiring additional genetic variation that you can see on the right-hand side of the slide. In later adenomas, a more profound collection of genetic variation as we move into a carcinoma under the microscope, and then acquiring the capacity to metastasize. And although this may sound, oh yeah, this is exactly what, you know, it sounds so simple today, it was actually extraordinary in 1990 and really launched a lot of energy to look at this more precisely, which of course we can today. And the picture is incredibly more complicated. So another really profound contribution to this area was, of course, Jeremy Jass's. This centre pathway was really Bert Vogelstein's traditional pathway of tumorigenesis. And Jeremy, again, another pathologist who took a lot of his ideas from what he saw under the microscope, started to pull apart colorectal cancer in terms of subtypes and really describe different pathways for different subtypes, depending on the precipitators of the neoplastic change, which Jeremy was fairly astutely aware of, given his interest in mismatch repair deficiency and Lynch syndrome. So analyses in the somatic space have, of course, benefited from the amazing technology advances that we've seen over the last decade or so. And I've skipped all of that to come to a figure from a recent Nature paper, which is really meant for effect, not for detail. But it is a report from the Pan-Cancer Group. It's used 2,658 whole genome sequences from cancer, a number of different cancers, as you can see listed across the top of this figure, and then detailed what genes have been identified to carry pathogenic variants in these cancers. And you can see the great variation. There are a great variation in types of genetic changes that are identified. There's a great variation in the frequency, and there's a great number of different genes involved in at least present in the tumour. And I know there's going to be some more discussion around which ones are actually fundamental to driving the tumour and which ones aren't, but nonetheless it's an amazingly complicated landscape. But just to get out of this as simply as I can, I just wanted to bring you to this part of the figure. And perhaps it's been a bit of a surprise to the community that a lot of this work is identifying a small proportion of variation that is germline. And this has really been helpful to us, I think, to move forward and to move cancer genetics, as Mendel knew it, out of the context of a family history and out of the context of early age onset, as Lee and Framini knew, and to realise that there is a portion of cancer, of all cancer, that actually is associated with a germline predisposition, which again is pushing us back to understand that there really is no great divide between our somatic and germline studies. And the more that we look at them together, I think the better that we're going to be to understand both predisposition and our tumorigenic pathways, and to be able to best manage individuals and advise individuals around genome sequencing outputs and even panel sequencing outputs. So thanks to Mendel, an amazing science blossomed. I think there's so much more to come, and I think integrating it into clinical care is really going to be our frontier for work into the future. I don't mean to say that what we've done has been not significant, but I think there's so much more to realise in tumour meetings and so forth that probably isn't being done at the moment, and I really look forward to being involved in that in the future. So thank you.
Fantastic. Thanks, Melissa. That was a great trip down memory lane, where we've come from and where we're going to. So thank you very much for your talk.